Hello, my name is Vipul Jain. I'm the founder and CEO of Advanced Cells Group, and it is an absolute pleasure for me to have you all with us today. Advanced Cells was founded with a simple yet very powerful vision to transform healthcare through the science of regenerative medicine. Over the years, we have grown into one of India's most trusted names in stem cell research and manufacturing. Our focus is purely on innovation, safety, and impact. From neurological conditions to orthopedic conditions, from organ specific diseases to age related disorders, we provide ethically driven, ethically sourced, ethically manufactured stem cells that are personalized to each individual. Our GMP compliant facilities are equipped with cutting edge technology, and our work is backed by a strong network of doctors, researchers, both in India as well as across the world. But what truly sets us apart is our commitment to the people, to patients, to caregivers, and to the medical community. At Advanced Cells, we don't just manufacture cells, we deliver hope, healing, and outcomes. Today's webinar is an important step in our mission to educate, calibrate, and move the conversation forward. We are excited to share the knowledge and explore what lies for regenerative medicine ahead. Thank you very much for being here. Let's begin.
I'm Meenu Jain, Director of Advanced Cells, and it is truly my pleasure to introduce today's speaker, someone who represents the very forefront of innovation in regenerative medicine, Dr. Maryam Asadi. Dr. Asadi holds a PhD in molecular medicine and a prestigious diploma from the American Board of Regenerative Medicine. In 2024, she was recognized with the Extraordinary Young Scientist Award by the American Society of Regenerative Medicine, an acknowledgement of her remarkable dedication, intellect, and impact in the field. Her passion lies in the regenerative medicine with a special focus on NK cell therapy. She has successfully secured a research grant for groundbreaking cellular cancer therapy project using CAR NK cells. Dr. Asadi currently heads the cell therapy department at ACECR and also works with Shiraz ACECR Hospital. In addition, she represents the American Board of Regenerative Medicine in Iran and is the founder and CEO of the Uni Health City platform, a bold initiative bringing innovation and accessibility together in the healthcare space. More than her accolades, what truly stands out is her unwavering commitment to bringing science closer to healing. Please join me in welcoming a truly inspiring mind, Dr. Maryam Asadi. The floor is all yours.
Really excited to share insight on a rapidly evolving and immensely promising area of regenerative medicine. I would like to express my sincere gratitude to Nilab for his dedicated effort in convincing me to participate in this webinar. I also extend my congratulations to Advanced Cell Company for fostering such a team of young, hardworking, and passionate individuals. Nilab's commitment to Advanced Cell is truly commendable. Today, you will show my slides, or I will do it myself. Can you see?
Yes, we can.
That's great. Today, we will delve into the fascinating world of exosomes, specifically focusing on the practical and therapeutic advancements that are shaping the future of regenerative medicine and precision therapies. I will guide you through the key aspect of exosomes from their fundamental biology to their application in these webinars. Okay. I want to be sure that everybody can see my slide. What are extracellular vesicles is title. Can you see?
Yes.
Okay. So let's begin. What are extracellular vesicles? To start, let's understand what we mean by extracellular vesicle or exosomes. Cells throughout our bodies constantly communicate with each other, and one of their primary methods is by releasing these tiny membrane-bound sacs. These fall into several categories: microvesicles, apoptotic bodies, and exosomes. As you can see, they differ significantly in size, with exosomes being the smallest, typically ranging from 30 to 150 nanometers. This size distinction is not just academic, it's profoundly impact how they interact with other cells and consequently their potential therapeutic role. Let's okay. Why cells release vesicles, exosomes have a very specific identity. They are characterized by the presence of certain surface markers, particularly tetraspanin like CD9, CD63, and CD81. These markers are crucial for identifying true exosomes, distinguishing them from other cellular debris or waste products. CD9, CD63, and CD81. What makes exosomes truly special is their cargo. They carry a complex payload of protein, various forms of genetic material, including DNA, RNA, and microRNAs, and lipids. They are encapsulated within a protective lipid bilayer membrane. This cargo is direct reflection of the parent cell, meaning the properties and therapeutic potential of exosomes are highly dependent on the cell type of their originate. As we can see, we have different cargo of exosomes from different source of cells. This slide shows what natural exosomes are made of and how they can be changed. On the left, you see a natural exosomes, which like a tiny bubble carrying things like fats, proteins, RNA, and DNA. On the right, it shows how we can put new things into the exosome, like special trackers or medications, or we can make it a different job, and such as delivery drugs jobs. Now, let's connect exosome to a major area of their therapeutic interest. Aging isn't just about chronological years. It's driven by a series of interconnected biological processes, often termed the hallmark of aging. This includes damage to our genetic material, shortening of telomeres, changes in gene expression, and issues with how our cell process proteins. As this hallmark accumulates, they contribute to the decline in tissue and organ function, manifesting as the various conditions we associate with aging. As I am looking at my desktop, I want to be sure that my voice and sound is okay. If there's any problem, I cannot see this screen of Meet. Because I'm focusing on my screen of PPT. Please, if there is any problem, just tell me by voice, okay?
Sure.
Could you please
Okay.
enhance the PPT? Could you see the option there?
Sure. Is it okay now?
Is there
Is it
any
okay?
option? Yeah, I think you should maximize
I maximize it. Can you see it?
Hi, Dr. Maryam. Hiba.
Can you see the maximize?
No, we need the presentation, PowerPoint presentation. Can you present it as PowerPoint presentation?
It is PowerPoint. I'm sharing
But a
my
slideshow.
PowerPoint.
We needed the slideshow.
Yeah, it is a slideshow, but I share my screen, and I think now I maximize it. You can see it. Am I right? Let's stop sharing and share it again. I think it's better to share PDF because it is bigger. Can you see?
Yes.
Is it
Yes.
better now?
It's better than before.
Okay. Okay. Now I'm explaining slide 8. Cellular senescence and its practice. One of the most significant hallmark is cellular senescence. I will close my camera. Okay. One of the most significant hallmark is cellular senescence. Imagine a cell that stops dividing, but instead of dying, it lingers, becoming dysfunctional. These senescent cells accumulate in our tissue as we age. Critically, they don't just sit there. They become pro-inflammatory, releasing a cocktail of molecules known as the senescent-associated secretory phenotype, or SASP. These SASPs contribute to chronic inflammation, tissue damage, and ultimately accelerate the development of many age-related diseases. Therefore, addressing cellular senescence is a key strategy in regenerative medicine. This is where exosomes enter the picture as a powerful therapeutic strategy. The goal is to either eliminate this detrimental senescent cell or more directly to reverse or delay the senescent process. Exosomes derived from young and healthy cells offer a unique biological tool to achieve this. By delivering their cargo of regenerative molecules, these exosomes can modulate the SASP, shifting the balance from harmful to beneficial. This fundamental mechanism allows them to promote a healthier cellular environment and counteract the effect of aging. Youngblood story is an interesting story. The idea that factors from younger organisms can regenerate older ones isn't new. It stems from outside, like heterarchical parabiosis, where the circulatory system of young and old mice are joined, demonstrating a remarkable reversal of aging in the older animal. It was so interesting that scientists found that transplant of young mice to a older make the older mice younger. Growing evidence suggests that exosomes are key mediators of these useful factors. When exosomes from young vibrant cells are introduced, they carry messages that can reprogram senescent cells and revitalize aging issues, uh, effectively slowing down or even reversing biological aging processes. Let's look a bit deeper at how exosomes achieve this. This rich cargo enables them to directly interfere with and suppress core component of cellular senescence. For example, they can promote the proliferation of healthy cells, reduce DNA damage, and dampen inflammation by modulating pathways like NF-kappa B. Critically, they suppress the secretion of those harmful SASP factors we discussed earlier, such as CXCL1, GDF15, GMCS1, and IL6. Molecular analysis confirmed that exosomes are rich in proteins that play vital role in regulating inflammation, immune response, and remodeling of the extracellular matrix and crucial for tissue health. Uh, Now we're going to know about importance of stem cell sourcing for exosome quality. Given that exosomes are a reflection of the cells that produce them, as we talked before, the source of the stem cells for exosome production is absolutely paramount. It dictates not only the therapeutic efficacy, but more importantly, the safety profile of the final products. We cannot overstate the importance of stringent ethical sourcing and meticulous screening of these donor cells. There have been instances in the past where importantly sourced or applied stem cells, like some mesenchymal stem cell therapy, have led to significant adverse outcomes. This underscored the need for robust control from a very beginning. This slide is about why picking the right source for a stem cell is very important for getting good, safe exosomes. How good and safe exosome products are depends entirely on where the stem cell comes from. Although it's really important to carefully choose and check donor cells to avoid contamination and make sure the treatment work well. Lastly, using a stem cell that weren't properly chosen or used can lead to bad results. The image here at the bottom shows different type of stem cell and their ethical consideration, including embryonic, fetal, and mesenchymal stem cell, highlighting that mesenchymal stem cells are considered ethically sourced, genetically stable, and have low tumor risk for developing exosome treatments. Uh, Okay. This slide explains that we are going to more detail in exosome application. In this slide, we show that exosomes have many different ways to help treat various diseases. Exosomes carry special signals that can affect many illnesses linked to aging, cells, and inflammation. Because they can help with so many different problems, exosomes are a very useful tool in medicine that help the body repair itself. The circle diagram shows how exosomes, labeled as the EV, can how this label can impact different conditions like lung disease, heart disease, and skin disease by either boosting or reducing certain biological process. Here again, we can see different application of exosomes, illustrating of downstream surgical application of exosome therapy. The therapeutic effect of exosome are a hierarchical translation through disease-specific tissue responses, tissue-specific cellular alternation, and cell-specific molecular signaling pathway. Here is a review on exosome application in clinical trials, perspective questions, and challenges. We can see COVID was the most prevalent application of exosomes before, and other application we can see here in clinical trials. Application in skin regeneration. In this photo, you can see one of our patients. One of the most immediate and visible application is in skin regeneration. Exosomes have demonstrated significant efficacy in cosmetic application, specifically targeting anti-wrinkle and skin firming effect. Research shows a measurable increase in collagen 1 content in cells treated with exosomes, leading to improved skin elasticity and reduction in the appearance of wrinkles. What impressive is that this maximum efficacy is observed at relatively low concentration, even surpassing the effects seen with traditional positive control. Here you can see before and after of our patients. Application in skin health, anti-inflammation. Beyond purely aesthetic benefits, exosome also offers substantial advantages for overall skin health through their potent anti-inflammatory properties. Studies have shown they can significantly reduce pro-inflammatory markers such as IL-6, which is often a key mediator in inflammatory responses. This indicates their strong potential for soothing irritated skin, managing inflammatory skin conditions like psoriasis, and generally promoting a healthier skin barriers. The dose-dependent effect reinforces their specific biological activity. I like to check again that there is no problem with the audience. Can you hear me, Bill?
Yes, loud and clear.
Okay, let's continue.
Yes, please.
Because I'm sharing my desktop, I can't see you well. So sometimes interval I will. Okay.
Hello.
Hello. I'm here.
Okay.
Okay. Example of exosome products. Currently, exosome products are being developed and made available in various forms to suit different application and delivery methods. We see topical formulation like facial serum and creams, typically containing billions of exosomes per container, designed for direct application. For more targeted or systematic effect, highly concentrated frozen exosomes or powder preparation are available, intended for medical and aesthetic clinics. These can be administered through subdermal injection, intranasal routes, or even intravenously, depending on the therapeutic goal. Okay. In this article, we know about the application of exosome derived from adipose-derived stem cell in skin wound healing. That is a review article. I bring just as a sample to know about exosomes, effects, safety, and efficacy. In this article, the writers have studied adipose-derived stem cell exosomes and their benefit in wound healing for skin regeneration. And they have a retrospective review of publishing different articles. Their results show that most studies have shown the possible role of adipose-derived exosomes in cutaneous wound healing through regulation of inflammatory response and promotion of cell proliferating migration and differentiation, angiogenesis, and matrix reconstructions. And we can see in this review article show that using exosomes in wound healing and skin regeneration have safety and efficacy. Okay. Here are stem cell derived exosome for the treatment of disease in orthopedic surgery and related specialty. It has a different source of exosomes, have different application in fractures, and we can see the result in femoral fraction and in different models, even human or animal model. It has also application in osteoporosis, umbilical cord mesenchymal stem cell, adipose-derived mesenchymal stem cell exosomes, and different source we can see in osteoarthritis, spinal cord injury, sciatic nerve injury, muscle and tendon tears, and different disc degeneration. Stem cell also have some application in, you can see here, in neurosurgery and ischemic stroke, for example, traumatic brain injury, different source of mesenchymal stem cell exosomes we can see in this column, and their different result showed safety and efficacy of this application. We have other application of exosomes in Alzheimer's disease and Parkinson's disease. Again, different source of exosomes. In MS, vascular dementia, and HIV, radiation-induced cognitive dysfunction, epilepsy, and other application in neurosurgery, even depression, stress, brain aging, and other application. Pardon me.
Sorry for interrupting, Maryam. Actually, your screen was not visible for last 1 minute. Can you just restart from there?
Okay, can you see it now?
Yes, now we can see.
We can see.
Okay. There is delay in showing the slide, I think. And there is clinical trial of exosome therapy in behavior and mental disorders, blood and lymph conditions, digestive system disease at or before birth, and eye diseases, heart and blood diseases, hormone-related diseases, musculoskeletal diseases, and neoplasms. There are different applications of exosomes in clinical trials. Nervous system disease, nutrition and metabolic diseases, skin and connective tissue diseases. I will share the slide with you after the presentation, so you can see the clinical trial NCT codes and you can search about them in detail in clinical trial gov websites. Okay. Here we can see the comparison of stem cell therapy and exosome therapy, their advantages and limitations. We have stem cell therapy with differentiation potential of cells, but we don't have disadvantages in exosomes. For stem cell therapy, we have it is applicable to treatment for a wide range of disease, but the range is less for exosomes. And also, we have the ethical issue limitation in stem cell therapy, immunogenicity, and infusion toxicity. In exosomes, although our limitation is insufficient regulatory control and not well distinguished industry standards, but they are relatively low yield for large scale manufacturing, and their storage is easier than a stem cell. Their delivery modalities is also better, and more concentrated functional cargo we can have in exosomes. And comparable therapeutic effect to a stem cell, their size is much smaller, but their effect is also compatible. But in exosomes, we must be sure about batch to batch inconsistently, and we have to be careful about quality control of each batch. Okay, we are talking about exosomes storage. How to store exosome so they last a long time. The main way to keep exosomes for a long time are freezing them deeply or cryopreservation, freeze drying them or spray drying them. For deep freezing, temperature and special antifreeze agent are crucial. Storing exosome at cool fridge temperature 4 degree Celsius can make them less effective. Instead, storing them at very cold temperature, minus 80 degrees Celsius, is best because it causes the least harm to their shape and what they carry inside. Finally, using a specific type of sugar-based antifreeze is the best option because it stops exosome from clumping together and getting damaged by freezing. Focusing on freeze drying and spray drying. Delicate material like exosome and vaccines can be easily stored by freeze drying them, and you can bring them back to life just by adding water. A recent study found that freeze drying with protective substances can keep the exosomes, important protein and RNA active for about 4 weeks, even when kept at room temperature. Unlike freeze drying, spray drying is a simpler one-step process, meaning you don't need expensive equipment or many steps. However, key things is spray drying, like how fast exosomes are fed into the machine, the spray pressure, and the temperature can all affect how stable exosomes are and if their contents stay safe. Uh, Comprehensive quality control and the value change. Now let's shift to critically important aspect, quality control, to ensure the highest safety and quality of exosome products. A robust and comprehensive quality control system must span the entire value chain. This begins with waste source, ethical and regulatory stem cell sourcing, including institutional review board approval and strict informed consent. It means that the company must be so valid and the company must have ethical consent. It then extends to rigorous stem cell screening for pathogens and genetic defects, followed by meticulous stem cell banking in CGMP compliant facilities to establish master and working cell banks. The next stage in quality control involves the manufacturing process itself. Exosome manufacturing must occur in certified good tissue practice (GTP) complement facilities, utilizing property and validate cell expansion processes. Most post-manufacturing strict cold chain logistics are essential for packaging and storage. Some companies provide good storage and shipment, typically at minus 80 degrees Celsius to maintain product integrity and viability. Crucially, each batch undergo extensive characterization using techniques like nanoparticle tracking analysis. I will show you in the next slide to precisely quantify particle concentration and size, and nanoflow cytometry to confirm exosome purity and identity based on the presence of key tetraspanin markers like CD9, CD63, and CD81. It is a SEM image. You can see the exosome particle size, and also some aggregation of them. Here also we can see their size and criteria. Finally, comprehensive safety testing is performed on every single batch to ensure the product is of medical grade quality. It is so important. Each doctor want to use exosomes must pay special attention to quality control tests and certificate. This includes a battery of tests for sterility, mycoplasma contamination, endotoxin levels, and cytotoxicity on various cell lines. The certificate performs irritations and sensitization tests for topical applications, as well as photocytotoxicity and oral systemic toxicity assay, all adhering to recognized international guidelines like OECD. Ultimately, strict adherence to good manufacturing practice or CGMP and good tissue practice GTP is not just a regulatory requirement, it's the cornerstone for ensuring patient safety and the consistent therapeutic efficacy of exosome products. Here you can see a certificate of a sample company about different criteria of their exosomes. We can see here the batch number, the date, date of manufacturing, release date. Mostly the manufacturing date and release date are different because the test needs some time to be released. If a batch released immediately to the market, it's not according to GMP and GTP regulations. And we can see sterility tests, mycoplasma tests, HIV, and other viruses tests that is done by real-time PCR and different. And at the end, the company must sign that they guarantee the information that is mentioned in quality certification. Thank you for your attention. And if you have any question, we are open to discuss about exosomes criteria.
Thank you, Dr. Maryam, for such an insightful session. I'm sure we've gained a lot of knowledge about exosomes and its applications. I would also like to invite Swati Sharma. She is from our customer services team. And so any questions, we'll be happy to answer.
Hi, good evening, everyone. Thank you, Meenu ma'am, for introducing me. Myself, Swati Sharma, and I have been associated with Advanced Cells for last 6 years. And I have seen many success stories in front of me. So I would like to invite everyone to have an interactive session about exosomes and stem cells. So please feel free to ask your questions.
I guess you've explained everyone so well, Dr. Maryam.
Thank you so much, Dr. Maryam.
Most welcome. I think somebody has a question.
Yeah, there is a question. You want to read it? Do similar types of tissues or cells produce different kind of exosomes? Can exosomes be synthetically replicated for specific therapies? Would you like to answer this, Dr. Maryam?
Sorry, I can't see the question. Do similar type of tissue cell produce different kind of exosome?
Yes. And can exosomes?
No. It is mirroring the tissue and cell that is exosome originate from. It is completely different, but mostly they use mesenchymal stem cell of different source, umbilical cord source or any other sources.
Someone has raised a hand.
Yes, it's me. I'm Professor Radom Hanna. I'm from Egypt and the Egyptian affiliate of the American Board. And also I'm the president of the Egyptian Society of Extracellular Exosomes. So I wanted to say a comment and also or 2 comments. The first one that through our society we do a monthly webinar all around the EV. So I would be, it's my pleasure to share the links with you so you can attend the webinar monthly about different subjects on the EV. This is the first comment. The second one is the clinical application for the EVs. I think we still do have some limitations or some obstacles that we need to cross first, like the standardization of their isolation, the mass production, the source, the doses. So all of this, though they are very promising non-cell regenerative tool, but for their clinical translation, there are some question marks that need to be answered first. What do you think?
Manufacturing procedures. I don't have your voice clear, but I think you asked about manufacturing procedure. It needs different sessions to talk about it, and it is related to producer, I think. But if you have any question, you can send me message directly in LinkedIn about each steps in manufacturing.
There is one more question, Dr. Maryam. Can stem cells be transplanted from animals into the human body?
It is an issue, that challenging issue. You know, we cannot transplant from animal to human in organ transplantation and cell transplantation. As exosomes also express the MHC and HLA, so it's not suggested to transplant exosomes from animal to human. Although, however, there is some articles that try to delete the HLA to omit the HLA from human. I mean, manipulating the animal exosomes can change it, and I cannot say it is safe, but it will decrease the alloreactivity. We have two kinds of transplantation, xenograft and allograft. Xenograft is from animals to humans, or from one species to another, but allograft is from the same species. Mostly, the transplantation from animal to human will be rejected. It can be organ transplantation, cell transplantation, and exosome transplantation.
So what potential do exosomes have in limb? Can I request everyone to mute themselves, please, so that we can go ahead with the webinar?
Yes. There is, we can explain it in orthopedic application. I showed some clinical trials NTC code that have some application in fractures and other regeneration. Yes, the answer is yes. We have safety and efficacy in limb regeneration also. What about allogenic transplantation? Can it be safe or it had to? Yes, allogenic is safe. Most of the exosome transplantation is allogenic. Even mesenchymal stem cell transplantation can be allogenic. Do you know why allogenic transplantation of mesenchymal stem cell and mesenchymal derived exosomes can be safe? Because mesenchymal cell doesn't express any HLA on their surface, and it is completely safe. Hello, Maryam. Good presentation. I have a question. How confident we are that when we purify the exosome, we don't have any non-exosomal and free-floating proteins and other biomolecular contamination? As some of our other friends asked about manufacturing, I didn't explain about manufacturing at all because it needs some more sessions. But according to your question, there is no floating proteins in the liquid, exosome liquid, but exosome itself can carry some proteins in their membrane, in their liquid membrane. Is there any other question? What potential do exosome have? No, it is repeated.
It is a very informative session, Dr. Maryam. You have presented that exosomes contain DNA. Will they have an impact on the recipient?
Sure, it has. It is genetic material, and it helps the recipient's regeneration.
Or any other karyotyping changes on the recipient?
Yes, and not impact changes, but they have healing efficacy. And DNA, RNA, and microRNA that is derived from exosomes, and they are derived from originate cell, have effects on the recipient. Another question, HLA expression on mesenchymal stem cell is according to the source. It's not expressed in CP-derived, but can be expressed from AT or bone marrow. Am I right? The question, Radwa, would you please explain more about your question?
Okay, I think it's better to talk. Can you hear me? What I'm saying that there can be some constraints in the allogenic transplantation of the mesenchymal stem cells or the derived exosomes. It's according to the source. So when they are derived from the cord blood, yes, the HLA is not expressed. But when they are derived from the adipose tissue, adults, or bone marrow, there are some HLA expression which can evoke immune system. So it's not always safe to take because you said the allogenic is totally safe from mesenchymal stem cells. So I'm saying it depends on the source of the mesenchymal stem cells. Not all mesenchymal stem cells and the EVs are safe for the allogenic transplantation. If the source is from the adipose tissue or bone marrow, we have to do HLA typing.
Yes. Thank you for your explanation. You are right. The source is also important, but in general, we can have allogenic transplantation of stem cells, mesenchymal stem cells. Mostly, they are the source that is used is umbilical cord source or placenta, or any other sources. Fat tissue and blood mesenchymal stem cells mostly used as a autologous source. Thank you for reminding, Radwa.
Thank you.
Okay. Or another question. Any other karyotyping changes on the recipient? No. It's not such a serious changes. DNA in the exosome cannot have karyotyping changes, but not serious change, not very important changes in karyotyping. Can a stem cell be extracted from a human dead body? We have a lot of source. Good question. We have a lot of source for exosomes and stem cell of fetus and mature human. So we don't need a human dead body. As you know, when the body is dead, the cell will also dead. But in some strike, we can get this cell, but we don't need to use human dead body. Okay, another question. Are exosomes more better than stem cells? We cannot say that. We cannot say exosomes are better in generally, but it is easier to use. You can storage easily exosome, but you cannot keep cell for a long time. Cell lifetime is mostly 6 hours after production, but exosome can be stored for more than 6 months. And about efficacy, it's a wide question. In different applications, the efficacy of exosomes and stem cells are different. You know, when we need long-lasting effect, it's better to use cells, stem cells. But when we need, for example, in some progressive neurodegenerative disease, stem cells is a sustainable source of producing exosomes in the body. But for regeneration, using exosome is easier. For orthopedics application, exosomes is easier to use for practitioners. Okay, another question. What disease can exosome potentially treat? I explained it in detail in my presentation. I will share the slide with you, but generally, the application is orthopedics application, neurosurgery application, regenerating application, different application in different subspecialty it has. Gyno application, bladder application. Okay. Another question, how advanced stem cells are better than other sources? Dear audience, I want to explain about this question. I'm not the promoters of advanced cells. Yes. This presentation is just a scientific presentation, and I will thank the Advanced Cell company.
I think Swati can answer that question.
Yeah. Thank you so much, Dr. Maryam. So I would like to answer this question, how advanced cells is better. So basically, to produce the stem cells, you have to have ISO certified and GMP certified labs. So advanced cells is into manufacturing and research of these specialized cells. So that is the reason we are the best in this field. And Dr. Sucheta has asked in what conditions we can use the stem cells, though Dr. Maryam has already answered the questions. So stem cells and exosomes basically can be used for various conditions. So if I talk about the neurological condition, so I will pick up few names so that everybody can relate with the diseases. So stem cells and exosomes can show very good improvement as far as autism is concerned, cerebral palsy, Parkinson's, motor neuron disease, ALS. So these are the neurological conditions where we have personally seen good improvement in the patients. If I talk about the orthopedic condition, then osteoarthritis, rheumatoid arthritis can be one area where definitely stem cell application has helped the patients. If I talk about anti-aging, which everybody wants to know about, so stem cells and exosomes have shown a very good improvement as an anti-aging agent, I would say, because it is having anti-inflammatory properties. So these are the areas where definitely stem cells and exosomes can help the patient.
Thank you very much for your detailed explanation. It's better to share your manual and brochures in this page to know audience more about your products.
Sure, Dr. Maryam, we'll do that.
Probably you can send a link to our website. I'll just put it here.
Okay, sure. Another question, what is the mode of administration when we are talking about any systematic disease treatment? Different route of injection we have. For systematic, we can use IV injection. And I think Advanced Cell has also IV injection source of exosomes. Am I right?
Yeah, that's correct.
Okay.
So basically, the route of administration will be disease specific. So as I discussed earlier, if we are looking at neurological condition, so in that case, generally the route of administration is intravenous and lumbar puncture. Of course, the physical evaluation of the patient will be required. That will be done by the doctor. But in general, if you're asking, then for neurological condition, we can give intravenous injection and lumbar puncture. If we are looking for orthopedic condition, then site-specific injection is very useful for the patient. Suppose anybody is having osteoarthritis, then you can give site-specific injection of mesenchymal stem cells and exosomes. And if you're looking for anti-aging, then IV injection works really well.
There's another question. What is the treatment process for orthopedic conditions at Advanced Cells?
So basically, our protocol is, I would like to guide you step by step so that if anybody is interested or if your relative is having this condition, since it's a very common condition, so you can just guide them. So I think the website link is already shared in the chat box. So you can share the medical reports of the patient with us, like MRI or X-ray and the medicines which the patient is already taking. We will be discussing the case history of the patient with our doctor, and then the patient will be called for the consultation. And now it is the patient's choice whether they want to go ahead with the stem cells or not. So the route of administration, as I said, would be your local injection to the knee joint or hip joint, or if it is rheumatoid arthritis, then it can be multiple joints as well, along with IV injection.
Thank you.
And generally, it's a daycare procedure.
Okay, thank you. Another question about autism treatment. For autism treatment, we can use both stem cells and also exosome together, or even a stem cell alone. It needs 3 doses of injection with 1 month interval. And if you want more detail about doses and sustainable effect, it's better to discuss more in my private LinkedIn page I introduced before in the first slide, I think. I think the time is over. I must go to another meeting. So I want to thank you, Advanced Cell executive team. I will leave the session. You can continue together. Enjoy your time and accept my warm attitude and gratitude to all your effort in conducting this session and webinars.
Thank you, Dr. Maryam. We are extremely grateful for you to spare this time and do the webinar for us. And it's an honor for us. Thank you so much.
Most welcome. Dear Menu Jane, I think.
Yes.
Yeah, thanks. And most welcome. Have a good time. Enjoy being with you. Goodbye.
Goodbye.
Thank you, Dr. Maryam. Thank you so much.
Yes. Thank you, everyone. Thank you for joining. Thank you. Bye-bye.
Thank you, everyone. Thank you.